hello, my name is Yaqueline Gutierrez, and today I'll be presenting my project on the role of the lamina mutation in cardiac conduction disorders, thanks to University of Houston Downtown and Baylor College of Medicine. Okay. So, a brief introduction. So, cardiac conduction disorders affect the cardiac conduction system. A little background information. Cardiac conduction disorders is essentially the heart's electrical system that's in charge of the heart's rhythm and its heart rate. So, as you can tell, it's very important. However, there's usually some problems that arise, and these are the cardiac conduction disorders, which technically cause irregular heartbeats and irregular heart rates. Because of this, many of the cardiac conduction disorders that we know of today can be life-threatening when they are left untreated. Some examples of some cardiac conduction disorders can be cardiomyopathy and atrioventricular block. A little bit of background: one of the cardiac conduction disorders can be seen through the lamina gene. So the lamina gene is a very active gene, and because it creates differential cells that are all over our bodies. However, for today's topic, we're going to be talking about the laminin AC proteins and some important roles that they play, which can be seen in gene expression, epigenetic regulation, and that sort of thing. So mutations in the lamina gene are present in patients that have cardiac conduction disorders, which is why it is important for us to look at them. Next. So the lamina mutation in our area of research of interest. So like I mentioned before, today's focus will be the lamina mutation, and this lamina mutation can be seen at R225X, which is in the chromosome. Understanding the molecular mechanisms of the lamina R225X are important because of its association with cardiac conduction disorders, which can lead to improvement in treatments in these patients. Next. So of course, like I mentioned before, many of these cardiac conduction disorders are in humans. However, we can't really test and do these experiments on humans, so we have to move to a mice model. So for example, right now we have this figure 1 with patients that actually carry the lamina 225X mutation. So essentially, we have a proband. Proband is essentially the first individual in a family that is seen with a genetic disorder. So using this figure, we can kind of use it to guide us to create mice models that can help us elucidate the mechanisms of cardiac conduction disorders in these mice. Next. So experimental design and mouse model established. So here we see a figure with figure 2, which is the mice model establishment. We can see a mouse with the mutation and a mouse that essentially allows us to look at the cardiac conduction system's reporter, kind of to see where the electrical system is being affected. So by breeding these two mice, we can have a mouse that can fit our mouse model. Essentially, we can do various methods on these type of mice, which could be surface ECG, telemetry ECG, EP, which essentially is the heart monitoring with X-rays, and echocardiography and immunofluorescence staining. Next. Right here is one of the results that we have, which is the immunofluorescence staining, which is confirmed to decrease lamina expression in the AV node of head mice. Right here in figure 3, we can see you have a lot of this red, which is basically the staining of the nuclear of the cells. While in figure B, there's a much more decrease of this red color, and this helps to show that the mice with the knock-in mutation show an exhibit of loss of function in the laminin AC protein. Next. In this slide, we have figure 4A and B. So in figure 4A, we can see that the mice show 2-degree and 3-degree of atrioventricular block. And this can essentially mean, like I said, is the irregular heartbeats and rhythms of the heart. And right here we can see that it's prolonged, which shows the irregular heartbeat. And right here we can see that the intervals become smaller, longer, smaller, and longer, which do show the progression of these cardiac conduction disorders. While in figure B, we see essentially the same thing with the help of the graph that shows that the wild-type mice, or the mice that have no mutation of the lamina gene, show that the incidence of atrial fibrillation is very low, while on the head mice, which are the mice with the mutation, show a significant increase in the incidence of atrial fibrillation. Next. However, when looking at the echo, which is basically looking at the inside of the heart for the contractions of the heart, we see essentially no difference. Here's the wild type and here's the het. You can see the structure of the heart and essentially there's no difference. You can't see a difference. The same can be said for figure 5C, which with the wild type and the het once again compared side by side, and there is no significant change in the contraction of the heart. The same can be said for all of these other graphs, where we can see that there is a bit of variation, but overall, the average of the heart contractions is relatively the same. So a conclusion is that the knock-in mice, the mice that have the mutation, do show a loss of function for the lamin AC gene, the protein, and these het mice also demonstrate current connection disorders. These means that there's an increase in atrial fibrillation susceptibility, which means that we are mimicking the diseases that patients carry in LABNA R225X mutations. So for future research, we can want to see the influence of pacemaker cells and current connection disorders, because how we saw in the echo, there isn't much change. So there has to be something else that's causing these cardiac connection disorders. And this is in relation to the development of cardiomyopathy relationship, because there is a lack of phenotype that is seen in the cardiac connection disorders but isn't essentially caused by the LAMNA mutation gene. And that's it. Thank you.